Right, thank you very much for the invitation to be here. So yes, I'm going to bring it back to kind of the early part of the lifespan. I may have fit better in the morning session, but I think as many of us have said today, I think that the kind of mechanisms that we're looking at that we see early in life probably have lots of relevance for things that persist throughout the lifespan. I'm also going to bring it back to the animal models. I am a basic neuroscientist. I am not an epidemiologist or a toxicologist, so I'll just put that out there right now. But hopefully this will be of interest to those of you who are. My lab is very interested in this concept of developmental programming, and I don't need to really explain what this is to this crowd. We're particularly interested in how events which impact the developing immune system can really have very persistent effects on health throughout the remainder of the lifespan. So, there's a saying that nothing in biology makes sense except in the light of evolution, and I like to change this to say nothing in biology makes sense except in the light of development. In other words, wherever you go, there you are. So what I mean by that, and again, don't have to explain it to this crowd, is that none of us just spontaneously arrives at a disease state or a phenotype as adults or later in life. We all get there through a kind of set of developmental trajectories and developmental experiences. And I think just kind of broadly, I think neuroscience in particular tends to forget that unless they are actually developmental neuroscientists. And there's a lot of evidence now, as you know, that neuroinflammation or inflammation during development can have a profound impact on the risk for disorders throughout the lifespan, and not just developmental disorders. And so while I will focus on autism today, of course, we think that these types of pathologies or pathological mechanisms that we're interested in can have impacts on even neurodegenerative processes much later. So why the immune system? Well, this has kind of a shared commonality with the interest in air pollution on the brain. The interest in the immune system in the brain is also quite new. So it's only in the past 10 years or so probably that there's really been this kind of explosion of interest in the role that so-called immune molecules, which of course we know are important for keeping us safe from infectious molecules or injury, are also really ubiquitously important for normal brain development. So we now know that all of these different factors, again, that have just been associated with the peripheral immune system for most of our knowledge of them, have critical roles that may be totally separate, that are actually important for just basic morphogenesis, neural differentiation, neuromigration, etc. And the resident immune cells of the brain, the microglia, have really come into the spotlight recently, not only in kind of neuropathology kinds of contexts, but also in just normal brain development. So these are really the focus of my work. They have been for many years now. And I'll tell you why we're so fascinated by these cells. Of course, they are the resident immune cells of the brain. They're primary immunocompetent cells of the brain. But they also play critical roles in normal brain development. So we know that they prune synapses. We know that they engulf apoptotic cells. They respond to neural activity. They probably do lots of things that we don't really even know about yet. So why are we so fascinated by them? Well, they are, in fact, the only cells that live in the brain that aren't from the brain. So they aren't derived from the neural ectoderm, but are rather derived during embryogenesis from the fetal yolk sac. And so this paper was published in Science in 2010. So this knowledge is only 8 years old, although it was suspected for a very long time that these kind of macrophage precursor cells from the yolk sac start coming into the developing brain starting in mid-gestation. And so they're really one of the very earliest arrivers to the brain. They're there before any other glial cells are there. And this is not specific to the microglia, so all of the macrophage populations of the body are seeded from the yolk sac early in development during this period of primitive haematopoiesis. But what makes the microglia unique and distinct from all the other macrophage populations is that the blood-brain barrier then closes. And so we now know, based on many studies, in mice at least, that in the absence of kind of overt trauma or a breakdown of the blood-brain barrier, you don't have any replenishment from the periphery of these initial precursor cells. So these precursor cells arrive into the CNS, they proliferate, and they set up shop. They become the microglia that then persists throughout the lifespan of the rodent. There is some turnover throughout life, but they can also be extremely long-lived. So this is quite interesting to us because it really suggests that they are especially susceptible to programming, persistent effects on their function when they're first seeding the brain and proliferating. So the question then, of course, is what implications does this have for neurological disorders? So a diverse kind of number of inflammatory events which activate these cells, and we know that based on many, many studies, while they're basically doing their homeostatic functions in brain development, it kind of brings up many interesting questions. And again, we have evidence from many animal studies now that if you have kind of diverse challenges such as infection, toxins, stressors, et cetera, that this can really impact the risk of disorders later in life. And now I have these kind of two separate trajectories here quite intentionally. I won't talk about this in detail, but I'll touch on what other speakers have mentioned this morning, which is the fact that there are sex differences in this process as well. There's sex differences in the way that microglia colonize the brain, and there are transcriptional differences in the response of microglia that are quite distinct between males and females. And in fact, female microglia mature more quickly than male microglia. So we think this may have some real implications for the types of disorders that we're all interested in, which tend to be sexually dimorphic. So autism, as you've heard about in detail this morning, this is a look at postmortem brain, which is one of several studies that came out several years ago now, showing that basically in the brains of individuals that died and were autistic, that even if you don't know anything about autism, you can see that the microglia look really weird. So they look reactive or dystrophic or whatever we want to call them. So this was kind of some of the first evidence that microglia might be involved in the pathology of autism. Of course, this is a postmortem study. It's always difficult to understand what comes first, and that really is I think the holy grail of these types of studies is, are microglia responding to neural dysfunction, or are they causing the neural dysfunction? And so for that, of course, we kind of move to animal studies. And so we're very interested in what are the environmental factors that may contribute to autism. And so what I'll show you in brief today is some evidence from our model in which we're exposing pregnant dams, not people, but mice, throughout pregnancy to diesel exhaust. And this combined with maternal stress during gestation has very persistent effects on microglia. It causes changes in synapse number and neural circuit function and behavioral dysfunction. And all of this is part of a very large collaborative project with my co-PIs at Duke University, Charlie Roglu and Kaf Sarasa. So I don't need to tell you, again, why we're interested in air pollution, and just to echo again what has been said is that the environmental contribution is at least as large as the genetic contribution, and it's one of the strongest environmental links to autism. So the model that we've been using is a diesel exhaust particle exposure model. And so we do these kind of tracheal installations throughout pregnancy. It's an intermittent exposure starting from E2 to E18, so really the whole span of pregnancy. I first started doing this work, which was is in collaboration with Ian Gilmore from the EPA, when I was still a professor at Duke, and this was part of the Children's Environmental Health Initiative there. It used to be led by Marilyn Miranda, and she kind of roped me into looking at this kind of project. And so all I will say about it, and the reason I got really fascinated by it initially, beyond, of course, the real-world relevance of it, is that it causes this persistent lung inflammation in the mothers, including macrophage infiltration into the lung of the mothers. And I thought that this was really interesting because some of you who may be aware of the animal literature on autism risk, there's a growing in popularity kind of model known as the maternal immune activation model, which typically involves a proxy for viral infection or influenza, which also begins in the lung. So I was kind of fascinated by this model that you've got this kind of persistent chronic inflammation beginning in the lung, and then what is the consequence of that for the offspring. And arguably, I think this model maybe approximates a larger percentage of the population, and when we're talking about the increasing risk of autism that we've seen. So one of the first questions we asked was, what is the impact on microglial colonization of the developing brain? And you've seen this kind of graphic before, where we just characterize the morphology of the cells from more round amoeboid. So this is, to point out, this is the kind of natural development of microglia. So when they first enter the embryonic brain, they're very round, they're very macrophage-like, and then over the course of normal development, they basically transition here into their more mature ramified form. So we simply asked, is there an effect of the diesel exposure on the normal distribution? And you can see that in the graph on the bottom there. And in fact, there was, but only in males. So in males, we see an increase at embryonic day 18 in the cortex of these round cells. No change in females. And there's no change in the total number of cells. So this is really a change in distribution. And this persists to postnatal day 30 at least, in which most of the cells are now this ramified mature phenotype, and we continue to see kind of more immature looking microglia in the males only. And we see this in cortex. We also see it in hippocampus, hypothalamus. I won't show all the data in the interest of time, but it's basically the same pattern. So we wondered whether this had any impact on cortical development. And again, this is really inspired by other models of maternal immune activation, which have been linked to autism-like phenotypes in mice. So for instance, with this proxy for influenza, this poly-IC model, this is data that was recently published. They show quite strikingly that there are basically patches of cells missing in the cortex just based on this single injection of a viral mimic during pregnancy. So we looked to see if there might be similar kind of morphological changes in the brains of our animals, and we really didn't see any kind of overt changes. So in general, the types of pathological changes we're seeing in response to the diesel alone seems to be much milder, for instance, in comparison to like the ultrafine particles that we heard about this morning. But one thing that the graduate student, Stephen, noticed when he was looking at these, and this is the value of really looking at your tissue samples, is that there seemed to be a change in the way that microglia were overlapping neurons. So he was interested in taking a closer look at this, and so using these very detailed kind of 3D reconstructions of microglia and neurons, he could really objectively quantify how much physical overlap there is between a microglial cell and a neuron. And this is just giving you a taste of that. So the yellow is the overlap, and then the software quantifies it for you, so it's entirely objective. And here we found something quite intriguing, and that is that only in males, again, there's more physical overlap between microglia and neurons. And this is really at the kind of cell body level. We don't know what this means yet. So I mentioned before, microglia could be responding to neural distress in some way. We know that they respond to neural activity, that we know that they respond to things like ATP. But it's intriguing, and especially given the male to female ratio, that there's larger effects in males. But what does all of this actually matter? So as was also presented this morning, autism is really primarily defined by behavioral changes. And so do we see any behavioral changes in these animals? And the answer is no. So we've really carefully phenotyped the behavior of these animals, and they're just perfectly normal mice as far as we can tell. But we weren't very surprised by this, and I already told you kind of a snapshot of this model at the beginning involves another factor. We know that, of course, hundreds of genes, dozens of environmental factors are linked to autism. Each alone only weakly predicts, and we've heard a lot about this today, there's multiple factors. So our interest is in whether multiple factors may be converging on inflammatory pathways to cause pathology. So essentially, what are the modifying factors for disease and why? And here again, we were inspired by the epidemiological literature. Rosalind Wright in particular, and many others now have shown that maternal stress, for instance, during pregnancy is a strong predictor of asthma outcomes in children. This is independent of exposure levels in the home. So this is really more reported levels of maternal stress being the stronger predictor. So we decided to incorporate stress into this paradigm. So again, just control or diesel exposure throughout pregnancy, plus or minus maternal stress during the last part of pregnancy. The stressor we're using is a model of resource deprivation, and this is a model of basically nest restriction, which was developed by Tally Barham at UC Irvine, and I think it's a really nice model of again, resource deprivation, which is something we were interested in trying to model. Tally's lab has shown really nicely that this increases stress reactivity in the mother and in the offspring very persistently. However, we decided to apply this prenatally as opposed to postnatally. And we did that for a couple of reasons. One is because we really wanted to restrict the types of exposure to the prenatal period because we were interested in autism, and increasingly we believe autism is of prenatal origins. And also, in our hands, this model, which simplifies things, we see no changes in maternal care. So once, right before the pups are born, the moms actually get back their full nest material and bedding, and they give birth, and then we really carefully scored maternal care, so they're caring for their pups just fine. And I think that's really important in the understanding of what might be going on. But what we do see is a true synergism now in terms of behavior. So we see no behavioral changes with either of the kind of hits alone, stress or diesel exhaust. But in combination, we see changes in communication or ultrasonic vocalizations, which is a proxy for communication. We see changes in social behavior. We see changes in cognitive behavior, anxiety. And I'll just show you a snapshot of those in the interest of time. So this is ultrasonic vocalizations, where we simply record the number of calls that they're making and look at the duration of the calls and the structure of the calls. And here, in both males and females, we see with a combination of diesel and stress that there's an increase in the number of calls, but the structure and the frequency of the calls is changed. And this actually persists into adulthood. So at least in male mice, what we did was we took them as adults and put them in a cage with an estrus female. And typically, males will start singing in response to this. And so we recorded their little songs. And we found something, again, quite striking. Hopefully, you can kind of understand this kind of plot. But basically, it's showing that the diesel stress males have calls in which the syllables are much simpler. So they have more kind of short, simple calls and fewer long, more complex calls. So we see this very persistent effect in communication. We also show that there are very pronounced social deficits, again in males. So this is already introduced before. So asking how much time do they spend investigating another novel mouse. And here you can see in males it's much reduced. And in fact, we had an object on the other side, in this case a rubber duck, and they were much more interested in the rubber duck. So taken together, the sociability score, the amount of social investigation, is not significant in our diesel stressed males, whereas our controls are very social. Females are social regardless. We also see changes in other social tasks, so like a social novelty preference task. So it's just a very consistent phenotype. And I will say too that this is really the most striking behavioral phenotype that I've seen in any of the models from my lab, and it's very, very reproducible, and always just a very striking difference between groups. So what's the impact of the combined stressors on microglia? And so we took kind of a gene expression approach initially to ask broadly what kind of transcriptional changes might be occurring in the cortex of these animals. And I won't show you all of the data, but what we found was actually pretty interesting, that we have kind of a selective change in these innate immune genes called TLR4 and IL-1 at postnatal day 30. And again, it's only in males that are exposed to kind of the double hit of stress and diesel. And this is an interesting couple of genes to change, because there's been a lot of talk about, you know, double hits. And here we think we may have actually a molecular mechanism by which this is occurring. So some of you may be familiar with the inflammasome cascade, which is a way that innate immune cells produce pro-inflammatory cytokines, pro-inflammatory IL-1 in particular, in which signaling at a toll-like receptor induces the production of the pro-form of a cytokine. And then a second signal is actually required to cleave this pro-form into its mature form. And so we basically have a coincidence detection system within a given cell type. And this then produces the mature pro-inflammatory cytokine. And indeed, we see that we have a massive upregulation of pro-inflammatory IL-1 beta protein within the brains, again, only of males exposed to the double hit of stress and diesel, and very low levels of IL-10, which is anti-inflammatory. And in females, we see the opposite. So we see high levels of IL-10, which we think is one reason they may be protected. We showed further using flow cytometry that this change in this pro form of IL-1 beta, which gets cleaved, is specific to microglia. So here we can look inside the microglia using flow to see, is it actually specific to this cell type, and it is. So, okay, so we clearly have a change in the inflammatory state of these cells, which seems to be this kind of double hit of priming and then a kind of activation signal. But what is this actually doing to behavior? We know that based on transcriptional studies by Dan Geschwin and other labs, that there are a couple of different sets of gene kind of families that are the most changed in autism. And those are genes that are important for synaptic function, which are downregulated, not surprisingly, and genes that are important for the immune system, which are upregulated, not surprisingly. So we wondered, you know, are we affecting synaptic numbers in this model? But then the next question we had was, where do we look? Which is not a trivial question, and this is actually one of the most challenging questions we've had to answer with this model, because autism could affect so many different brain regions, and probably does. But based on the literature, one region we've really been kind of focusing on is the anterior cingulate cortex. This is a region that's critical for all of the kind of behavioral changes that I've talked to you about. And there's a very large literature in both human and animal models that there are changes in this brain region. And in particular, we were interested in the fact that reduced thalamocortical synapses have been reported. So these are these projections from the thalamus, which are VGLUT2 positive, and they project into the synaptic zone of the anterior cingulate cortex. And so we can measure these specific kinds of synapses using a marker called VGLUT2, and then we can also compare it to a marker called VGLUT1, which measures these intracortical synapses, intracortical connections, which are not thought to be as implicated in autism. And so we use an immunohistochemistry approach where we can basically look at the co-localization of the pre- and post-synaptic marker and ask, how many synapses are there. And what we found was that we have specifically a decrease in the number of these VGLUT2 positive synapses, so these thalamocortical synapses that project. And this is at postnatal day 15 that I'm showing you, but we have recent data now showing that this decrease in the number of these synapses persists until postnatal day 100. So we know that it persists into mature adulthood at least. We don't know how long they persist, but it's probably a permanent change. So are there effects on microglia in the ACC? So there are no changes in the number of microglia, and in fact in all of our models we never see a change in the number, and even if you do it on a layer-specific basis. But what we did find was quite striking in this area is that, so this is labeling microglia with two different immunohistochemical markers. One is called P2YR12, it's a purinergic receptor, and one is called IBA1. And these are really widely considered to be constitutive markers for microglia, meaning that they're always expressed and they can kind of go up or down, but they're always there. Well, we first looked at the number of microglia using just IBA1, and here this is again the value of really looking at your tissue. The graduate student working on this noticed that it looked like there were actually holes in the tissue when she was just using a marker for IBA1. Long story short, what she found is that the cells were actually labeling for just one of these markers or the other. And so they're completely dropping their expression of IBA1 and upregulating P2YR12, for instance. And you can look through like a column of tissue and really see these very striking patches of the anterior cingulate, almost evocative of that study showing patches or cortical patches in that maternal immune activation model. And so she quantified this, and indeed what she found is that basically this diesel stress combo is just increasing the heterogeneity of microglia in this brain region. So it increases whether they're likely to express one of these markers or the other, which is very strange. This has not been reported in the literature ever. So we're still kind of trying to figure out what this means. But what we know is that it's transient, so it kind of goes up at postnatal day 8, high at P15, and then comes back down. And we also know that early in development, this kind of heterogeneity in microglial gene expression is increased, but it decreases as you mature. And so we're wondering if basically all of this is telling us is that it's a delay in the maturation of these cells. And then finally, are there functional differences? So we decided to say, well, if you look at a cell that has equal expression of these markers versus just one or the other, is there a change in their phagocytic capacity, so their ability to engulf debris and degrade it? And we can do that using a measure called CD68. And indeed, what we found is in these very heterogeneous microglia, which express only one cell type, one marker or the other, they have this decreased phagocytic index, suggesting that they're not as good at clearing as other cells. So altogether, what we found is that we have this specific decrease in these thalamocortical connections in ACC of our 2-hit kind of diesel stress males, and this increase in microglial heterogeneity. And finally, in just the last couple seconds, or not seconds, last minute, I'll just touch on some data that's in progress, but we're excited about it, which is to ask, okay, so if you have synaptic changes in one brain region, like the ACC, then how does this change behavior? How is it affecting now the whole brain? So is it changing the way the circuit works? And so for this, we're using this multi-unit recording. So this is the brilliant work of Kavsarasa at Duke, who basically puts these electrodes into the brains of awake animals and records simultaneously from 10 different brain regions at the same time. And he's recording from mice as they're interacting with another mouse or as they're interacting with an object and saying, you know, what does a normal social circuit look like and what does an abnormal social circuit look like? And can you use the circuit to essentially identify what the mouse is doing, whether they're interacting with a social stimulus or an object? And he's done this approach basically using machine learning, and I won't go into all the details of this because I probably don't understand it, but basically the take-home here is that you're looking at the coherence of how different brain regions are kind of talking to each other. And he's used this approach effectively to basically predict whether an animal will be susceptible to depression or not, based simply on how the circuit is working. And so we asked the same question. So can we look at the circuit of these mice and say, you know, are they interacting with a social stimulus, another mouse or an object? Can we distinguish between them just based on this circuit activity? And here's our control animals, both males and females, and the kind of machine learning approach is doing a pretty good job of predicting it because they're significantly greater than chance, so suggesting that they are distinguishing between a social stimulus and an object, whereas both our male and female diesel stress animals are basically not significantly different from chance. So they're not distinguishing between an object and a social stimulus. So just looking at their circuit and brain activity, we can tell which group they're in, which is kind of amazing. So we're hoping to now use these data to understand better what part of the circuit is actually broken. So we can start asking more refined questions about, okay, so if we see synaptic changes in one brain region or another, what's the most important brain region and when is it happening? And of course, are microglia actually engulfing those synapses differentially and using kind of single cell sequencing to try to get at that question. In conclusion to all of that, we know that this kind of maternal immune activation through these combined factors produces this very striking behavioral, cellular, and circuit phenotype. And we're hoping that our ongoing work will really determine how microglial activation, so what is it about that heterogeneous phenotype at a single cell level could ultimately result in some targets to try to intervene. And excitingly, there's actually a lot of drugs that target the inflammasome pathway in very specific ways, and they're blood-brain barrier penetrant. So there's a lot of things kind of on the market that we could think about being able to give and specifically inhibit some part of that inflammasome pathway that seems to be reacting to the second hit of stress. So this is obviously a very large group effort, so I tried to highlight the people that did the work along the way, and I also just want to point out that this work is mostly funded by NIEHS, so thank you.